Okay, we'll proceed now with the next speaker. This was mentioned earlier to us by Dr. Sanchenes, si Dr. Jayvee Saco from the University of Batangas State University, Batangas City. So, Dr. Jayvee.
All right, thank you, Ting.
So, okay, you may proceed, please.
All right, so magandang umaga po sa inyong lahat and thank you for coming on joining us to this session. And thank you, Ting, for introducing me. Kinakabahan ako bigla. Well, good morning, everyone. So I'll be presenting to you about green tide blooms, its implication to coastal ecosystem, and possible research opportunities. So good thing Fred already introduced about the general perspective about seaweeds, and now we will try to do focus on on single on a specific species. So specifically on green tide bloom. So I would like to introduce also myself from the 2-year-old Marine Research Center of the Batangas State University, which is called Verde Island Passage Center for Oceanographic Research and Aquatic Life Sciences, for VIP corals. All right. So green tide blooms is caused by overproliferation of cosmopolitan green seaweeds such as ulva, ulvaria, and other ulvophyceae that produce blooms. So actually, these species are leaf-like, sheet-like, or filamentous-like in morphology and has thin and fleshy in texture. So in recent years, this is one of the major environmental problems that the world is facing. It has been observed worldwide from the continent of Europe in Ireland, France, and Netherlands, and to North America in the USA, and in Asia in Korea, China, and even in the Philippines. So actually, this was highlighted during the Beijing Olympics in 2008 that almost hinders the hosting of the sailing regatta competition. So actually, it covers about 600 kilometers square that lasted over 2 weeks, which it took more than 10,000 people to clean up and remove over a 1 million ton from the coast and beach area. So under optimum conditions such as excessive light nutrient, highlight conditions, and increased temperature, the bloom will occur and greatly associated with eutrophication. So what are the top sources of nutrient pollution leading to eutrophication? So according to Ocean Health Index, these are the sources of eutrophication, such as municipal sewage, agricultural fertilizer, livestock waste, stormwater drainage, and aquaculture. So to summarize the dynamics of eutrophication to marine environment, this diagram show that when there is an excessive nutrient runoffs plus increasing surface water temperature will produce algal bloom, then later on algae will decay and sink in the bottom, leading to the depleted oxygen layer, thus killing other marine organisms. So furthermore, these green tide blooms greatly affect commercial leisure fishing, human health and tourism activities, and even threaten coastal biodiversity. So in the Philippines, this actually became very controversial and widely publicized during the 2017 incident on the island of Boracay. So under normal circumstances, actually, this green tide blooming species are usually and really observed during summer season or dry or hot season. However, years before the 2017 incident, and on my observation with a frequent visit on the island for recreation purpose, it was observed that these species were present year-round and not only during summer season and already producing bloom. So as you could see on the pictures, these are some of the pictures taken on the island during the 2017 incident. So this leads to the closure of the whole island for rehabilitation by no other than the President of the Philippines. So, however, not to disrupt the tourism activities in the island, some group published an ad, something like this, to justify the occurrence of green tide bloom in Boracay. And they said that this species, um, that the species causing green tide is what makes the sand of the island. But it is actually not the truth because the sand of the Boracay came from um, a calcified seaweed, which is Halimeda species, and not from a fleshy seaweed, which is the ulva that causes green tide blooms. So the last time I visited the island was March 2019 and July 2019. The rehabilitation seems to have a significant improvement in the island with little and few traces of green tide blooming species. However, this could happen again and again because until now we don't have scientific data, sound investigation that could elucidate why they produce bloom. So moreover, this could happen also to the rest of the coast of the Philippines as there is increasing industrialization, tourism, and human activities, and even climate change. So, although there are some studies in green tide blooms in the Philippines, but merely focus on taxonomic identification, initial reproductive biology, and growth experiment, however, there is a very few studies done on determining their ecophysiological responses that might provide insights into the mechanism that cause these green tide blooms. So it is by understanding the photosynthesis and growth of these green tide blooming species under different physical-chemical parameters and their relationship could provide us information on elucidating the cause of green tide blooms. So, but why study their photosynthetic response? Because for seaweeds to grow, they need to utilize light energy in converting water and carbon dioxide to oxygen and carbohydrates respectively in the process called photosynthesis. So what can we do to elucidate the green tide blooms? So these are some of the scientific methods such as photosynthetic measurement by oxygen evolution technique using photosynthesis regions curve and pulse amplitude modulation fluorometry, as well as growth response by culturing from isolated zooids and distribution profiling of green tide blooming producing species. So these are some of the scientific methods that could be able to determine possible answers on the occurrence of green tide blooms in the Philippines. So first method directly measures one of the byproducts of photosynthesis, the oxygen. So these are actual pictures of my experimental setup wherein we could control all physical chemical parameters such as irradiance, temperature, salinity, and others. So this is the graphical representation of the measurement wherein it shows here that when irradiance increases, photosynthesis increases as well. But at a certain point where no matter how you increase irradiance, photosynthesis remains the same. So with this, it provides insights on their productivity, efficiency in light in utilizing low light um, for photosynthesis and their photochemical strategies, photoaccumulation strategies. So secondly, by measuring the activity of PS2 or photosystem 2, which is the actual site of light-absorbing chlorophylls together with PS1 by pulse amplitude modulation or PAM polarimetry. So this is the picture of the actual experimental setup where we could also control different chemical parameters as I've stated above. So this is the graphical representation of the measurement that could yield more information on their photosynthetic responses. So I've divided here dark adapted state and light adapted state. So at dark adapted state, the site of photosynthesis is relaxed. And when a strong light is given, it fully closes. So at this point, we could measure their tolerance to stress. Then, at light adapted state, the site of photosynthesis is very active, and when a strong light is given, it fully closes again. So at this point, we could measure the efficiency of light absorption, and at the end of the measurement, we will have a far red, and we could measure their photoprotective responses. Also, doing surveys on assessment of the distribution of green tide blooming species will provide information on the changes on the cover of the species in the specific area. So the cover will imply how much area the species inhabit the coastal zone, thus would provide predictive capacity on when and where a possible green tide bloom will occur. Then aside from studying the growth under different physical-chemical parameters using isotadzoids from green tide blooming species, we could positively utilize them by culturing for different purposes. So we could use them as food for human consumptions, such as rice toppings and snacks. Also, as a fertilizer, face mask, and anti-aging cream, and for pharmaceutical purposes, for antimicrobial properties, for treatment of cancer, for prevention of sexually transmitted disease, for preventing heart diseases, and even antiviral property, and actually a lot more to be explored. So with that, I am ending this presentation and thank you for listening and for your time. And I would like to acknowledge my host institution, Batangas State University, my host institution for my Balik Scientist program, and DOSTP Art for the Balik Scientist Award. So thank you very much and magandang umaga po sa inyong lahat.
Thank you very much, JB.
Yes.
Uh, Nobody yet posted a question for you. Would you like to add something to your presentation or expound something, a topic in your presentation?
Okay, so actually at this time we already submitted a proposal for studying this green tide bloom species. Actually, this is in collaboration with Dr. Wilfred Santianes of UPMSI. So we will try to elucidate the phenomenon of green tide bloom. So that includes most of the methods that I've presented here, and actually we are very open for collaboration. And I have some information that UPV is already developing a technology for culturing this green tide. I mean specifically ulva, and we are very much welcome for collaboration with them in terms of technology for culturing this type of species.
Is there someone raising his her hand?
Wala po, sir.
Okay, that's good. But
Okay.
if you are but if you are interested on doing such research topic, you may contact me if you want to do collaborative work for this research. And I would also like to siguro advertise our DOST funded research project entitled Marine Biodiversity Assessment in Verde Island Passage. And one of the goal of the project is to determine which marine resources, specifically seaweed, seagrass, and coral, are in Verde Island Passage. So with this one, we could able to map different seaweed resources in the whole Verde Island Passage.
Okay. Thank you very much, Dr. Saco. Wait, there's a someone posted on the Q&A.
I think it's from
Can you?
Ma'am
Can I ask
Georgie.
you the
Yes. Can you?
Yeah, sige.
So can you just?
Yeah, yung question ni Ma'am Georgine, why don't we invest more effort in setting up more value adding in seaweed raw material instead of exploring only semi-processed? Can I ask this? Hi, can I ask this further? Okay, siguro, um, I'm not sure how could I answer this, but actually the university, the Batangas State University already doing some effort on doing value adding to seaweed raw material. Like for example, actually I'm one of the thesis panel for that um, of that research, and we actually used crude extract of Kappa-ficus on doing biofilms, and also we use sargassum to fortify some of the um, baking products of that being created in our university. So I'm not sure how to answer this, but But since we are still developing technology on producing this commercially important seaweeds aside from the Capaphycus and Eucuma, we really need to have biology and physiological study first so we could able to produce them efficiently. And siguro later on po, ma'am, we could able to do value adding for the seaweed resources.
Very quickly lang, can I?
Yes, ma'am. Go ahead.
Yes. First, seaweeds number 1 in volume natin na aquaculture, but number 3 or 4 in value.
Yes po, ma'am.
And then, recommendation ng previous speaker, more research on more species and technologies, ganon. And yet, other countries like Malaysia is producing a yogurt, mango flavored, and then ang major raw material, carrageenan, which is most probably from the Philippines. Ang point ko lang, aside from studying more species, yung mga grow out technologies, marami na. You go to UP, BFAR, and CIFDEC, ang dami na, no? Why don't we invest more effort dun sa downstream? And this is maximizing the value added, no? Kasi it's Malaysia and it's the other countries that are using our semi-processed products and making money, a lot of money. Tayo hanggang semi-processed. Why can we not put up those factories, no? Yung mga yogurt, kung ano man yan.
Yes,
those that give the value, and this will require hindi lang academe, but actually engaging government, no?
Yes,
Governance na yan. So,
kaya actually, ma'am, yung project namin is closely, if ever ma-approve siya, it will be closely in collaboration with our Food Innovation Center in our university, in Batangas State University, because we already did some preliminary studies on doing value adding to these marine resources. And they're actually doing a nice result for this because I'm part of the thesis panel of some of the students doing preliminary studies on this, ma'am.
O sige, yung na hook up na with DOST, DTI, yung no?
Yes po, ma'am. Sige po.
Pwede man mag-research more species, more biology, more ecology, pero other countries are already making use of the technology that we have developed. Ano ba? Let's open our eyes. Yun lang.
Yes po, ma'am.
Thank
Take
you.
note of that, ma'am. Thank you very much, ma'am.
Okay.
Thank you, Ma'am Georgine. Thank you, JB.
Thank you, Ting. Ingat kayo dyan.
Congrats. Okay. So,